students, today we'll talk about muscles and muscular tissue. And let's revise the classification of all tissues. You already know that tissues are classified into bodyline tissue, it's epithelial tissue which provides border between inner and outer environments, and inner tissues. We have tissues of inner environment or connective tissues, which include blood and lymph, connective tissue proper and skeletal tissues, and also we have special types of tissues or specialized tissues which provide special features, special functions, and they include muscle tissue and nervous tissue. So they have some specific properties, that's why those tissues are specialized. And today we'll talk about muscle tissue. So muscle tissues, they include different types, and all types they are characterized by contractility, its ability for contractions. It's common feature for all types of muscles. And this contractility is provided by presence of actin or thin filaments and thick filaments or myosin filaments. So all types of muscles, they have actin and myosin, special filaments, special fibers which provide contractility of muscles. And they are necessary for contraction, actin and myosin filaments. So they are basic and they are common for all types of muscles. And there are three basic types of muscle tissues, and you should know them. There are smooth muscles or smooth muscular tissue, cardiac muscle or cardiac muscular tissue, and skeletal muscles or skeletal muscular tissue. And now let's talk about them, about those types, and at the end of our lesson we'll come to the classification of those types of tissues. So we will begin from smooth muscle tissue. Here we can see a diagram which shows smooth muscle tissue, and let's look what cells are located in this type of muscles. We can see here basic cells of smooth muscles, they are called smooth myocytes. Here we can see two neighboring smooth myocytes, and they have spindle-like shape. They are elongated, and they have elongated oval nucleus inside and cytoplasm. Here we can see them, so smooth myocytes. And they are distinct cells. They have cell junctions which provide communication between neighboring smooth myocytes, and also they provide connection between them. Here we can see shape of this smooth myocyte. It's spindle-like shape, and there are neighboring cells, and they are attached to each other side to side. Here we can see two sides, and another smooth myocyte is attached to this cell side to side. And here we can see a pattern of localization of arrangement of smooth myocytes in smooth muscles. Here we can see numerous smooth myocytes and pattern how do they form smooth muscular tissue. So one smooth myocyte, its structural and functional unit of smooth muscular tissue, and numerous smooth myocytes, they form smooth muscular tissue. And they are arranged in the layers. Here we can see a layer of smooth myocytes, and between layers of smooth myocytes we have thin layers of loose connective tissue with blood vessels and nerves, which provide nutrition and protection, and they fill the spaces between smooth myocytes. So here we can see this structure. And here we can see that smooth myocytes, they form layers. Here we can see a layer of smooth myocytes, and they undergo contraction at the same time. All smooth myocytes of the layer, they have contraction of at the same time. And smooth myocytes can be arranged in different layers. And here we can see longitudinal section of smooth myocytes. We can see their spindle-like shape. And here we can see layers of smooth myocytes, and between them there are layers of loose connective tissue with blood vessels and nerves. And here we can see smooth myocytes in the longitudinal section, And here we can see cross section, transverse section of smooth myocyte. We can see nucleus, cytoplasm, and sections in these places. And here we can see appearance of smooth myocyte in the transverse sections. Here we can see it. So here we can see longitudinal and transverse sections of smooth myocytes. And appearance of smooth muscular tissue at the transverse section is like this. So we can see sections in the middle part of smooth myocytes with visible nucleus, and peripheral parts of smooth myocytes. Here we can see them without nucleus, because those parts of the smooth myocytes, they don't contain nucleus. So it's appearance of smooth muscular tissue on transverse section. And different organs, they may contain some layers of smooth myocytes. They are arranged in different directions, and on the sections in histological slides we can see longitudinal layers and transverse layers of smooth myocytes. So here we can see transverse section of smooth myocytes and longitudinal section of smooth myocytes. We have made section of them, and they have different orientation according to the axis of the organ, and that's why they have different appearance according to the axis of the section. So here we can see longitudinal and transverse section of the smooth myocytes. It's diagram. And some organs, they have two layers. For example, in the gastrointestinal tract there are two layers of smooth myocytes in different directions. Sometimes there are three layers in some organs, and during the special histology classes we'll study what organs do have smooth muscles and how they are arranged in them. So here we can see longitudinal and transverse section, and let's look at this drawing. Here you can see smooth myocytes, a spindle-like shape. It's longitudinal section of smooth myocytes, and it's transverse section of smooth myocytes. Two layers, neighboring layers, and between them there are thin layers of loose fibrous connective tissue. Here we can see fibroblasts of this tissue, and also here we can see nerve cells. It's autonomic nerve ganglion, which provides regulation of the functions of this smooth myocytes. So here we can see appearance of smooth muscular tissue under microscope. And let's look at the microphotograph of smooth muscles. Here we can see smooth myocytes, the longitudinal sections, and here we can see them. It's layer of smooth myocytes and their appearance under microscope. And here we can see also smooth myocytes with their spindle-like shape. It's longitudinal section of smooth myocytes. Here we can see transverse section of smooth myocytes. It's bundle of smooth myocytes, and some of them contain nuclei at this level. Some of them don't contain nuclei because it's another part, it's peripheral part of the cell, and nucleus is somewhere but not on this section. And here we can see also smooth myocytes. There are nuclei, violet nuclei, elongated, and cytoplasm is pink. Here we can see their appearance. Also, longitudinal section of smooth myocytes, elongated cells with elongated nuclei, and transverse section of smooth muscular tissue. Here we can see them, and layer of loose connective tissue. And here we can see transverse section of smooth myocytes and longitudinal sections. And here we can see this light structure. It's autonomic nerve ganglion between smooth myocytes. It provides their regulation. And here you can see artery. We will study arteries, and this artery, it has numerous smooth myocytes. It has muscular tissue. It forms muscular layer, and it provides contractions of the arteries. It's necessary for their functions, to provide their functions. And at the microscope we can see a layer of smooth muscle inside the artery, and it's necessary to provide contractions of the artery. And here we can see microphotograph of the artery. Here we can see also smooth muscles. So what are the places of localization of smooth muscles? So first of all, there are blood vessels, arteries, and here we can see vein. which also contains smooth muscles in the middle layer. They are necessary to provide their contractions, to provide their functions. And also smooth myocytes are present in smaller blood vessels. For example, it's arteriole, the smallest artery, and here we can see transverse striations. There are nuclei of smooth myocytes. They also are present here, and here we can see them. Also, many internal organs, they have smooth muscles. All organs of gastrointestinal tract, tubular organs, they have smooth muscles in their wall. Organs of reproductive system, female and male, organs of urinary system, respiratory system, they have smooth muscles. So smooth muscles are of two types, two basic types. They are vascular or they are visceral. Vascular smooth muscles, they are present in blood vessels. Visceral, they are present in hollow internal organs. For example, it's esophagus. Here we can see longitudinal sections of smooth myocytes, and here we can see layer of smooth myocytes on their transverse section. Here we can see this organ. And microphotograph of the esophagus, this muscular layer here. It's transition of the esophagus into stomach, and here we can see muscular layer. Also, it's stomach, and here we can see muscles inside the wall of stomach. And here we can see this thick muscular layer. It's ureter, and also here we can see two layers of muscles. It's muscular layer of ureter, and microphotographs shows us muscles in the middle layer. It's urinary bladder with muscles inside the wall of bladder, and here we can see smooth muscles inside the wall. It's lung, and there are bronchi and bronchioles, and in their wall we also can find smooth muscles which are necessary for contractions. And here we can see smooth muscles in the wall of the small bronchi. It's uterus, and also it has muscular layer which is necessary for the contractions. And here we can see in the uterus smooth muscles. It's epididymis and tubules. It's organ of male reproductive system, and tubules of this organ are surrounded with smooth myocytes. And here you can see microphotograph, and they surround these tubules. It's prostate gland, and also smooth myocytes are present here. Almost all organs, many internal organs, almost all hollow organs, they contain smooth muscles, and also blood vessels, they contain smooth muscles. So two basic types of smooth muscular tissue, vascular type and visceral type, but they have common features of structure, and that's why we describe them as single type of muscular tissue. So what are the features of smooth muscles? They are composed of smooth myocytes, and also they have no striations. They are unstriated. The cytoplasm of smooth myocytes doesn't have visible myofibrils. That's why we call this type of tissue non-striated. And also smooth muscular tissue is involuntary. We cannot control this tissue. We cannot send the impulses to our stomach or to our arteries to provide their functions. That's why this type of tissue is involuntary. Let's go to the next type of muscles. It's skeletal muscle tissue. Skeletal muscle tissue is those tissue which forms muscles which are attached to our skeleton. They move our body. And skeletal muscles, first of all, they are voluntary. They are those muscles which are under our mind control, so we can control them, and they are skeletal muscles. They have bigger volume than smooth muscles, and they move our arms, our legs, they move our body, so they are skeletal muscles. And now let's look at microscopic structure of skeletal muscle tissue and skeletal muscles. So here we can see structure of skeletal muscle, and basic structural unit of skeletal muscle, it's muscle fiber. Here we can see muscle fiber. smooth muscles are composed of smooth myocytes, and skeletal muscles are composed of muscle fibers. Here we can see them. So let's look at the structure of muscle fiber and what is it. Here we can see cells. They are progenitors of skeletal muscles. They are called myoblasts. They derive from myotome of somites, and here we can see distinct cells. And later, those cells they are developed in cells progenitors of syncytium, myosyncytium, and then they are fused. Here we can see those cells are fused. They provide formation of rows, and here we can see that cells are fused. Here we can see them. Neighboring cells are fused and they form syncytium. It's postcellular structure or supracellular structure. It's syncytium. It contains numerous cells. It derives from some pre-existing cells when they are fused. And here we can see syncytium. So it's supracellular structure. And in muscles we call this myosyncytium because it belongs to muscles. So it's myosyncytium. It's result of fusion of myoblasts. And then this myosyncytium is enlarged in volume, and nuclei they move to the periphery of this myosyncytium. They usually are located under the membranes, because in this place, in the central part of the myosyncytium, myofibrils develop special cell organelles which will appear here in the central part. So what is myosynplast? It contains its supracellular structure, and it derives from some pre-existing myoblasts, and there are some specific terms which are characteristic for myosynplast. It's sarcolemma, sarcoplasm, sarcoplasmic reticulum, and myofibrils. So here we can see terms beginning from sarco. Sarco, it means meat, and everything what begins from sarco, it has relation to muscles. So membrane of myosynplast, cell membrane of myosynplast, also may be called sarcolemma. Plasmolemma of myosynplast also is called sarcolemma. Cytoplasm of myosynplast also may be called sarcoplasm. Here we can see sarcoplasm. It's another name of cytoplasm of myosynplast. Also, we have sarcoplasmic reticulum. Sarcoplasmic reticulum, it's modified endoplasmic reticulum of muscles. It's a smooth endoplasmic reticulum of muscles, and it's called sarcoplasmic reticulum. And also there are special organelles which are called myofibrils. They are very important for muscles, and they are special organelles. You know that there are general and special organelles. General organelles are present in all cells, and some cells they have special organelles. So myofibrils, they are special organelles which may be found only in the muscles. So they are characteristic for muscles, and they are present inside the myosynplast. So here we can see structure of myosynplast. A tubule, which is made up of some neighboring cells. There are numerous nuclei. Sarcoplasm, and sarcoplasm is surrounded with sarcolemma. And nuclei are numerous because this structure derives from some pre-existing cells. And what are the special organelles? Myofibrils. Here we can see myofibrils. They are present inside this muscle fiber. They are arranged in the bundles, and they provide contraction of the muscles. They are essential for the muscles. Here we can see a myosynplast, and inside this myosynplast we have myofibrils, bundles of myofibrils, and presence of myofibrils provides transverse striations of the skeletal muscles. Here we can see transverse striations, dark and light discs or dark and light bands in the myofibrils are visible. And why do we have those bands, light and dark? Because there are special contractive filaments, special contractive proteins which provide this pattern. So here we can see pattern dark band, light band, dark and light band. And let's look at this band. So what do we see here? Here we can see light and dark part and light again. And this part of the myofibril, which is the smallest unit of the myofibril, is called sarcomere. So it's sarcomere and it contains different proteins. So it's structural and functional unit of the myofibril. Later we'll talk about arrangement of sarcomere and how does it provide the contraction. But you should know that skeletal muscles as well as cardiac muscles, they have myofibrils which are composed of sarcomeres, and that's why those muscles, they are striated. They have transverse striations in their cytoplasm. But smooth muscles, they haven't these sarcomeres, they have another type of muscle contraction, and that's why they haven't transverse striations. So here we can see electron microphotographs of the sarcomeres. Here we can see transverse striations, dark and light regions, and also here we can see dark and light regions and bundles of myofibrils, and it provides formation of transverse striations. And here we can find numerous mitochondria between those myofibrils. Here we can see them. There are numerous mitochondria of the skeletal muscles. And on the smaller magnification we can see muscle fiber and numerous bundles of myofibrils with transverse striations. And this is nucleus of the skeletal muscle fiber. Here we can see it, it's located under the sarcolemma. And it's light microscopy of skeletal muscles, and here we can see transverse striations. So we can call this type of muscles also striated skeletal muscles. And here we can see myofibrils, and between them there are numerous mitochondria. Why do we have numerous mitochondria in the muscles? Because we need a lot of energy for muscle contractions. That's why there are numerous mitochondria. They provide synthesis of ATP. Also, we have sarcoplasmic reticulum, which is necessary for contractions. Here we can see myofibrils, and between them there are tubules, branched tubules, which surround each single myofibril, and they contain something. Sarcoplasmic reticulum is modified endoplasmic reticulum, smooth endoplasmic reticulum, and you know that one of the functions of smooth endoplasmic reticulum that it's storage of calcium ions in the muscles. So exactly, sarcoplasmic reticulum is storage of calcium ions which are essential for the muscle contraction. And what are the systems in the sarcoplasmic reticulum? It includes two systems of the tubules. Here we can see sarcolemma, cell membrane of the muscle fiber, and there are T-tubules which are beginning from sarcolemma, and electric signal runs alongside T-tubules inside the muscle fibers. There are T-tubules. And also there are terminal cisterna. They haven't connection with covering sarcolemma, but they are present inside. Terminal cisterna, they contain calcium. Here we can see terminal cisterna, they contain calcium, and they are neighboring with T-tubules. Here we can see one T-tubule and two terminal cisterna on two sides, and we call this triad. Here we can see triad. And when muscle fiber undergoes a signal that it's necessary to provide contraction, electric signal runs on the sarcolemma, it enters inside the muscle fiber, goes through T-tubule, enters in the sarcoplasm, and sends the signal to the terminal cisterna, which surround this T-tubule. And terminal cisterna, they release calcium ions, which are going to the myofibrils. It's necessary for the muscle contraction. So sarcolemma gives rise to T-tubules, and each T-tubule is surrounded with two terminal cisterna, and it's called triad. It's sarcoplasmic reticulum, specific reticulum, endoplasmic reticulum of the muscles. So we have muscle fibers. They include myosyncytium. It has sarcolemma, sarcoplasm, numerous nuclei, myofibrils, sarcoplasmic reticulum. But muscle fibers, they include something else. They also include myosynplast, and also they include myosatellite cells. They are present here, or satellite cells, and also it includes basement membrane. Here you can see satellite cells. They provide regeneration of muscle fibers. When myosyncytium is broken, is damaged, satellite cells may replace damaged area, and they provide regeneration of the muscles, of skeletal muscles. That's why regeneration of skeletal muscles is possible. And also satellite cells together with myosynplast are covered with basement membrane. Here we can see basement membrane. It covers myosynplast and satellite cells. So they have common basement membrane. It's membrane which covers muscle fiber. So together we can call this muscle fiber. It includes myosynplast, satellite cells, which are covered with basement membrane. Here we can see myoblasts or myoprogenitor cells, which form, some of them form myosynplast, and some cells, they aren't included in the myosynplast. Here we can see that some cells, they undergo transformation, they form myosynplast, they are fused, and they form myotubules, which are a result of cells fusion, and then we can see myosynplast. And some cells, they aren't included in the formation of myosynplast. They are neighboring with myosynplast, but they are not included in this formation. And they later are surrounded with basement membrane, and we know them as satellite cells. So satellite cells, they have common sources of the origin with muscle fibers, with myosynplast, but they are not included in the formation of myosynplast, and later they may be included because they are some kind of the stem cells. So satellite cells, they are stem cells of the muscles, of skeletal muscles. And here you can see basement membrane which covers this muscle fiber. So there are some stages of the development of skeletal muscles. Here we can see that there are stage of myotome cells, myotomes of somites, from myoblasts. Myoblasts, then they form muscle tubules. Muscle tubules, they form myosynplast and satellite cells. And here we can see mature muscle fiber. And there are satellite cells, and it's myosynplast. And let's look at the organization of muscle as an organ. Here we can see muscle fiber, and there are numerous muscle fibers, neighboring muscle fibers, and they are arranged in the bundles. Here we can see bundle of muscle fibers, and here we can see numerous muscle fibers. Usually we don't see satellite cells, we usually see only nuclei of muscle fibers, and there are numerous neighboring fibers, and the thin layers of loose connective tissue, they are present between the muscle fibers. Those places aren't empty, and we call those layers of loose connective tissue, we call them endomysium. They provide nutrition and innervation of the muscle fibers because they contain blood vessels and nerves which are going here to provide innervation of skeletal muscles. And here we can see bundle of muscle fibers, neighboring fibers, and layers of loose connective tissue between them are called endomysium. And here we can see a drawing of histological slides. There are muscle fibers. Here we can see on the longitudinal sections, cross sections, and here we can see that nuclei are in the peripheral parts of the muscle fibers, and thin layers of loose connective tissue. Between single muscle fibers we have endomysium. And here we can see muscle fibers on the light microscope. And let's compare skeletal muscles and smooth muscles. Smooth muscles are made up of smooth myocytes, single cells, and skeletal muscles are made up of skeletal muscle fibers. And they form bundles, and in the smooth muscles they form layers. And here we can see appearance of skeletal muscles under microscope. Here we can see muscle fibers, longitudinal and transverse section, and between them there is thin layer of connective tissue. It's called endomysium. It's present here. And also here we can see muscle fibers on the longitudinal sections. Here we can see also, and transverse striations are clearly visible here. Here you can see tongue, histological slides of the tongue, and also here you can see skeletal muscle fibers in different directions. And tongue, muscles of tongue, they are the strongest in our body because in the ratio of power till each individual muscle fiber. So it's the strongest muscle. And here we can see muscle fibers. Here we can see them that they are surrounded with connective tissue. And here we can see muscle fibers and they are surrounded with connective tissue. It's called endomysium. Here we can see that different muscle fibers are stained in different shades. It's because of presence of different types of muscle fibers. We have two basic types of muscle fibers. They are fast glycolytic fibers and slow oxidative fibers. Fast glycolytic fibers, they are light or white fibers, and slow glycolytic fibers, they are slow oxidative fibers, they are red or even brown. Because they have different ways of ATP synthesis. In fast glycolytic fibers, we have a lot of glycogen inclusions because we need glycogen for synthesis of ATP via glycolysis. And they don't need a lot of oxygen for the generation of ATP, and they can work in a condition of hypoxia, of deficiency of oxygen. And that's why they are light, they are white, because they don't contain a lot of pigment myoglobin. Slow oxidative fibers, or red muscle fibers, they contain a lot of protein myoglobin. It's a protein which is like hemoglobin of our blood. It provides red staining, red color of slow oxidative fibers. Why do we have hemoglobin or myoglobin in the muscles? We have myoglobin because it's storage of oxygen. Oxygen is necessary for the ATP synthesis in these muscles because Krebs cycle is involved in the ATP synthesis. So those fibers, they are working slowly but during long time because they need a lot of oxygen. They have storage of oxygen. It's myoglobin which provides red staining of those fibers. But fast glycolytic fibers, they are light because they don't contain or even don't contain myoglobin. They contain glycogen inclusions and they can work in a condition of hypoxia. So, like sprinters and skiers in sports, we have two types of muscles. Light or white muscle fibers, they are working very fast, but during short time, and even in condition of hypoxia. Red fibers, they are working for a long time, but slowly, like skiers. And different muscles, they have different ratio of those muscle fibers, but almost all our muscles are mixed. They have both types with different proportions, and we need both types of muscles. So here we can see muscle fibers. Dark are red fibers, they are slow and they work during long time, and light, they are fast and they are working during short time. Let's go to organization of the muscle as an organ. You know that there are muscle fibers and they are organized in the bundles. Here we can see muscle fiber, and between muscle fibers there are thin layers of connective tissue which are known as endomysium. Here we can see endomysium layers. And it's bundle primary bundle. Between some bundles of muscle fibers, there are layers of connective tissue, loose connective tissue, which are known as perimysium. Here you can see perimysium, which surrounds muscle bundles, bundles of muscle fibers inside the muscle. Perimysium also contains nerves and blood vessels. They are bigger than those which are present in the endomysium. Endomysium includes very thin layers, and perimysium it includes bigger layers. And entire muscle is covered with epimysium. It covers entire muscle. It also contains blood vessels and nerves. All those endomysium, perimysium, epimysium are made up of loose fibrous connective tissue with blood vessels and nerves. And also entire muscle is covered with dense fibrous connective tissue which forms fascia. Here we can see fascia, and some muscles they have aponeurosis. They are attached via tendons to bones, and those structures are made up of dense fibrous regular connective tissue. Here we can see a region of dense connective tissue here and here, white regions. They are made up of dense fibrous connective tissue. Here you can see endomysium, very thin layers inside the muscle bundles, and bigger layers are perimysium, and entire muscle is covered with epimysium, and epimysium is covered with fascia. Here we can see endomysium and numerous muscle fibers in skeletal muscle. And here we can see endomysium inside the skeletal muscle. Here we can see muscle fibers, bundle of muscle fibers. Inside them there is endomysium, and bundle is covered with perimysium. And here we can see blood vessels which are necessary to provide nutrition of this muscle. And here we can see muscle bundles inside endomysium, and between them there is perimysium, and entire muscle is covered with epimysium. Also a bundle of muscle fibers and layers of connective tissue between them. And entire muscle is covered with fascia, dense fibrous connective tissue which is called fascia. And tendons, they provide attachment of muscles to bones. Here you can see them. They are made up of dense fibrous regular connective tissue. Here you can see muscle fiber and transition of muscle fiber into a bundle of collagen fibers of tendon. And here you can see muscle with striations and its tendon, skeletal muscle and its junction to tendon. And also there are nerves, there are branch nerves inside the muscles. They send their branches to different muscle fibers, and you should know that each individual muscle fiber receives innervation from different nerves. So each individual muscle fiber has individual innervation. It has own source of innervation. And here we can see that they have separate innervation. But in smooth muscles, smooth myocytes, they form layers, and entire layer has same source of innervation. That's why contractions of skeletal muscles, they are more specific than in the smooth muscles. And also there are sensory structures. There are muscle bundles, muscle spindles. Here we can see its sensory structure, which reacts to the tension and distension of the skeletal muscles. It reacts to work of the skeletal muscle. It's located inside the muscle, and it provides sensory information, and it sends it via nerves to CNS. Let's go to the cardiac muscular tissue. So, cardiac muscle tissue is made up of cardiac myocytes. You remember that smooth muscular tissue has smooth myocytes, skeletal muscles. They have skeletal muscle fibers, and cardiac muscle has structural and functional units. They are cardiac myocytes. So there are individual cells, not myosin cytomes, there are individual cells, but they aren't the same with smooth myocytes. Here you can see elongated shape of the cardiac myocyte. It's cylindric in shape, but also it may have branches. It may have branched shape. So let's look at the structure of these cells. They have communication with neighboring cells, and there are intercalated discs. Here we can see intercalated discs between neighboring cardiac myocytes, and they provide communication between neighboring cells. Here we can see intercalated discs, and there are gap junctions or which provide communication between neighboring cells, or they are called nexuses. And also there are desmosomes which provide mechanical attachment of the neighboring cells. And here we can see also myofibrils and mitochondria. So let's look how they are attached to each other. Here we can see cardiac myocyte, and there are neighboring cardiac myocytes, and they are attached to each other end to end. In smooth muscles, attachment is side to side. In the cardiac muscle, end to end. And they form these rows of the cardiac myocytes, end to end attachment. And intercalated discs are present here. There are desmosomes and gap junctions which provide mechanical attachment and communication between neighboring cells. Via gap junctions, electric signals and irritation, excitement may pass through entire row of muscle cells and entire cardiac muscle. And here we can see intercalated discs between neighboring cardiac myocytes. There are desmosomes and gap junctions, and also adherent junctions are present here. It's intercalated disc. And here we can see myofibril and also cardiac myocytes. They have sarcoplasmic reticulum, which is very similar to those in the skeletal muscles, and myofibrils are present here. And there are mitochondria, numerous mitochondria, which also are necessary to provide energy for the cardiac muscle. Feature of the cardiac muscle is presence of nucleus inside the sarcoplasm of the cell, not under the cell membrane, inside the sarcoplasm. It has central or a little bit eccentric localization. And we can distinguish skeletal and cardiac muscles on the cross sections using this feature, using this tip. That nucleus in the cardiac muscle it has central position. And we can compare skeletal and cardiac muscle. In skeletal muscle, nuclei are located under the membrane. In cardiac muscle, nuclei are present inside the cytoplasm, in the central position or a little bit eccentric. And another feature, in skeletal muscles, we have triads in sarcoplasmic reticulum. In cardiac muscle, we usually have dyads, one T-tubule and one terminal cisterna, or they don't form this clearly visible triad as in the skeletal muscles. So let's look at the organization of skeletal muscle. Here we can see a cardiac myocyte and attachment of neighboring cells end to end. And here we can see row of these cells and neighboring rows. They have the same arrangement, but we can see connection between neighboring rows via anastomosis. Here we can see anastomosin and branching structure of the cardiac muscle. There are rows of the cells, and neighboring rows have communication via anastomosis. So we describe structure of cardiac muscle as anastomosin and branching structure. And here we can see pattern of the organization of the cardiac muscle. Here we can see that all cells are connected with each other. Why do we have this network in the heart? Because we need contraction of all cardiac myocytes at the same time. You know that there are two phases of the work of heart. There are systole and diastole. During contraction, it's systole. All myocytes should be contracted, and during diastole, they are relaxed. So This connection, this network, provides communication between all cells, and it results in the contraction of all cells at the same time. It's necessary for the work of the heart. And here we can see there are distinct cells. Its main difference with skeletal muscles, because skeletal muscles, they contain muscle fibers which have lost borders between neighboring cells, but here we have borders between neighboring cells. So under microscope, if you see intercalated discs, you can distinguish skeletal and cardiac muscles according to the presence of these intercalated discs, and also branching structure. Skeletal muscles, they don't have these branches, lateral branches, and communication between neighboring fibers. And here we can see structure of the cardiac muscular tissue. And also there are thin layers of the loose connective tissue between cardiac myocytes. They provide nutrition, protection, they provide innervation. There are capillaries between cardiac myocytes which bring here oxygen, which is necessary for the work of heart. And here we can see a drawing of histological slide. There are cardiac myocytes, there are intercalated discs between cardiac myocytes, and there are branches, anastomosis between cardiac myocytes. It's anastomosis and branching structure. Here we can see longitudinal section of the cardiac muscle and its cross section of cardiac myocytes. They may have different orientation of myocytes, and here we can see that nuclei, they have central position in the cardiac muscle, and its endomysium with fibroblasts, and also here we can see blood vessel which brings here oxygen. And microphotograph of the cardiac muscle. Here we can see intercalated discs and cardiac myocytes. It's nucleus and endomysium between cardiac myocytes. And here we can see cardiac myocytes, it's cardiac muscular tissue, intercalated discs, and nuclei of those cells, and layers of loose fibrous connective tissue, it's endomysium. So let's compare cardiac and skeletal muscle. Skeletal muscle, it's made up of muscle fibers, distinct fibers which have no connection with neighboring fibers. In cardiac muscles, there are functional muscle fibers or rods of the cardiac myocytes, but they are distinct cells, they aren't fused, but they have communication with neighboring rods. Its first feature, so here we can see distinct fibers in the muscles, in skeletal muscles, which have no borders between the cells. It's syncytium and individual cells in the heart, but they form functional fibers, and those functional fibers, they have communication with neighboring fibers, functional fibers, and they form anastomosing and branching structure. They form network of the cardiac myocytes which have communication with all neighboring cells. And in the skeletal muscles, we have innervation of each individual muscle fiber. Also, a feature of the cardiac muscle is involuntary, and skeletal muscles are voluntary. You know that skeletal muscles are under our mind control, and we cannot control cardiac muscle. We cannot send signal to provide contraction of our heart. It's involuntary. So here we can see these features. And another feature that skeletal muscles, they have satellite cells which provide regeneration of skeletal muscles. But cardiac muscles, they haven't satellite cells, they haven't another stem cells, and that's why regeneration of the heart or of the heart muscle is impossible because there are not stem cells. And when heart muscle is broken, for example, after cardiac infarction, some cells they die, and those damaged places in heart may be replaced only via indirect regeneration via replacement by fibrous connective tissue. It forms fibrous connective tissue scar inside the heart. But regeneration of skeletal muscles is possible because of presence of satellite cells which provide regeneration of skeletal muscles. And now let's compare cardiac muscle to smooth muscle. In smooth muscle and cardiac muscle we have individual cells, not myocytum as here. We have individual cells. Here smooth myocytes, here cardiac myocytes. Here they have connection site end to end, and in smooth muscles they have connection side to side. Here they form layers. Here they form an anastomosis and branching network-like structure. And also regeneration of cardiac muscle is impossible. Regeneration of smooth muscles is possible because some stem cells which are common for fibrous connective tissue and smooth muscles, they provide regeneration of smooth muscles. What is common for both types? They are made up of distinct cells, and both types of muscular tissue are involuntary. Skeletal muscle is voluntary, but cardiac and smooth muscles are involuntary. We cannot control their functions. So all inner organs except heart, they have smooth muscles, hollow internal organs. But heart, myocardium of heart, it has cardiac muscle, cardiac muscular tissue. And here we can see pattern of organization of cardiac muscular tissue and microphotographs of this cardiac muscle. And here we can see intercalated discs between the cells. That's why we can distinguish this muscle and skeletal muscle. Here we can see drawing of histological slide of heart wall, and here we can see numerous cardiac myocytes in the wall of heart. And here we can see anastomosis and branching structure of the heart muscle. It's cardiac muscular tissue, and its connective tissue, it's endomysium of heart. So now we are going to the classification of the muscular tissue. We have discussed all types of muscular tissue, and now let's compare them. Here you can see three basic types of muscle tissue. Here we can see smooth muscular tissue, cardiac, and skeletal muscular tissue. Smooth, cardiac, and skeletal. Three basic types. So, according to the presence of striations, we divide all types of muscular tissue into non-striated or smooth and striated. Here you can see non-striated and striated. Smooth, it's non-striated, and cardiac with skeletal, they are striated muscular tissues. So it's first classification. We classify muscular tissue into non-striated and striated muscular tissues. Non-striated it's smooth, striated it includes cardiac and skeletal. According to mind control, smooth and cardiac aren't under our mind control. Skeletal muscular tissue is under our mind control. So smooth and cardiac are involuntary, skeletal muscle is voluntary. It's another feature, another classification, involuntary and voluntary. Involuntary includes smooth and cardiac muscles, voluntary it's skeletal muscle. According to structural and functional units, smooth muscular tissue, it's made up of smooth myocytes. Cardiac muscular tissue, it's made up of cardiac myocytes, and skeletal, it's made up of skeletal muscle fibers, so it's myosyncytium. So smooth and cardiac, they have cells. They are composed of individual cells. Skeletal is composed of symplasms or myosymplasms. They are supracellular structures. It's another feature of classification. Smooth and cardiac composed of cells. Skeletal is composed of symplasms. And here we can see three basic types of muscles. Structural and functional unit of smooth muscle, it's smooth myocyte. Of cardiac muscle, cardiac myocyte. Of skeletal muscle, it's skeletal muscle fiber. So now we are going to contraction of striated muscles, skeletal and cardiac muscles. Let's look how they are contracted. So you know that muscle tissues and their contractility is provided by presence of actin, thin filaments, and myosin of 6 filaments. They are necessary for contraction. So contractility is provided by two types of filaments, actin and myosin filaments, and they are arranged in different patterns, in different ways. And let's look how do they provide muscle contraction. Here we can see sarcomere. It's structural unit of the myofibril. You know that there are myofibrils, which are special organelles of muscles. And here we can see part of myofibril which is called sarcomere. So let's look how thin and thick filaments are arranged. So here we can see two parts, two borders of the sarcomere. They are called Z-lines. Z-lines because this line has Z-like shape, two Z-lines. They are borders of the sarcomere. In the center of sarcomere we have another part. It's central part of sarcomere. It's called M-line. So central part, it's midline of sarcomere, it's M-line. We have thin filaments which are called actin filaments. They are attached to Z-lines. Here we can see on both sides of Z-lines we have actin filaments. And also we have thick filaments. Here we can see they are shown in red. They are myosin filaments. They are attached to M-line. So try to remember that actin filaments, they are attached to Z-line, and myosin filaments, myosin, they are attached to M-line. And they have longitudinal orientation, and there are some regions where only actin filaments are present, only myosin filaments are present, or both types of filaments are present. And we can distinguish some discs in the sarcomeres. So here we have light discs where actin filaments are only present. We call those discs or bands I-band or isotropic bands. They are light under microscope because only thin filaments are present here. And here we can see a disc where myosin filaments are present. Those discs or bands are called A-bands. Here we can see A-band or anisotropic band. It's dark band. Where myosin is present, those regions are dark. And here we have orientation and arrangement of bands in the sarcomeres. I-band, A-band, I-band, A-band, and they follow each other. I-band includes actin only, and A-band, it includes myosin. It corresponds to region of myosin presence. And also we have small region near the midline, near M-line. It's H-band. H-band corresponds to those region, light part of the dark band, where myosin only is present. So it's H-band inside A-band. So probably it's difficult to remember from the first time, but try to understand. I-bands, they are light, they are isotropic, they are light because they have actin only. A-band, anisotropic band, it's dark because it contains myosin, and it's darker when actin and myosin are present on the lateral sides, and near midline it has a little bit lighter band. It's called H-band, where myosin only is present. It's arrangement of sarcomere. I-band, A-band, I-band, A-band, and inside A-bands there are H-bands. Borders of sarcomeres are Z-lines, and midline of sarcomere is M-line. And now let's look what happens to sarcomere during contraction. Here we can see orientation of sarcomeres in the myofibril, and here we can see that dark bands, A-bands, they are located between I-bands, between light bands. So A-band, I-band, A, I-band. Dark, light, and here we can see that why do we have transverse striations in the muscles, because of presence of those bands in the myofibrils. And here we can see light regions in myofibrils, they correspond to actin regions, and dark, they correspond to myosin regions. Little bit lighter regions inside A band, they are H bands, regions where myosin only is present. And here we can see Z lines, I bands, A band, and M line. Here we can see M band, mid part of the sarcomere. Here we can see Z lines. It's I band where actin only is present, and A band where myosin is present. Light band, dark band, and inside light band we have Z line. Inside dark band we have M line. And also here we can see dark and light bands, and it results in the transverse striations of the skeletal and cardiac muscles. Both types, they have this arrangement of actin and myosin filaments. And what happens to them during contraction? Here we can see myosin filaments and actin filaments, they form common area, and here actin filaments are attached to myosin filaments, and myosin filaments, they move actin filaments to M line, and actin filaments are moving Z lines, and it results in the shortening of the sarcomere. So here we can see interactions between myosin and actin filaments, they move actin filaments to the M line, and Z line, they are moving to M line, and sarcomere is shorter. And during relaxation, they are going to their previous localization. Again, myosin filaments, they are moving actin filaments to the M line, and sarcomere is shortened, and it results in the contraction of the muscle. And relaxation. Contraction, and H band almost disappears. I bands, they are thinner, they become to be thinner, and A band is the same, but because length of myosin filaments is fixed. And then relaxation. It's what happens during the muscle contraction. Contraction and relaxation. So it's resting sarcomere, it's stretched and contracted sarcomere, and here we can compare them. Z lines, I and A bands, and H band almost disappears during contraction, because Z lines, they are moving to M line, and sarcomeres, all sarcomeres of the myofibrils, they are shortened, and it results in the contraction of the muscle. And how do myosin filaments do communicate with actin? They form cross bridges which are attached to the actin. There are active centers on the actin, and they are moving actin filaments to the center of the sarcomere via this way. So myosin filaments are attached to the actin filaments, and they are moving actin filaments to the M line. How do they provide this connection? Here we can see actin filament, and active centers of the actin, they are closed by troponin molecules. They close, and attachment of myosin filaments to the actin filaments is impossible. And also we have troponin complex. It includes troponin I, troponin C, and troponin T, 3 parts. Troponin reacts to the presence of the calcium ions. When calcium ions are released, troponin complex removes tropomyosin from actin, and myosin can be attached to the actin. Let's look. It's troponin complex. Calcium ions are released, and they remove tropomyosin from active centers of actin. And now attachment of myosin to actin is possible. And now active centers of actin are free, and myosin heads may be attached to the actin. After this, calcium ions, they move tropomyosin from active centers of actin, and myosin heads may be attached to the actin. Where do we receive calcium ions? Calcium ions are released from the sarcoplasmic reticulum. When nerve impulse comes here to sarcolemma, it runs through the T-tubule, goes to the muscle fiber, to the myofibrils, and here triads, terminal cisterna, they contain calcium ions which are released to the myofibrils. Here we can see nerve impulse, it comes here and it's distributed on the sarcolemma of the muscle fiber, and then calcium ions are released from sarcoplasmic reticulum, and after that they are going, here we can see T-tubule, terminal cisterna, which release calcium ions after this irritation, and calcium ions, they are attached to Troponin Complex, which removes tropomyosin from active centers of actin, and attachment of myosin heads to active centers of actin is possible, and it causes contraction of muscles. Here we can see. So troponin reacts to calcium, removes tropomyosin from active centers of actin, and myosin heads are attached to the myosin binding sites, active centers of actin. And ATP is necessary to provide this attachment. Myosin heads, they release energy via hydrolysis of ATP, and it's needed for fourth generation. And after that we have flexion of myosin heads, they release energy, and new molecule of ATP is attached. It's necessary for reattachment of the myosin heads from actin, and it's cycle of muscle contractions. ATP is attached to the myosin, it receives energy from ATP, attaches to the actin, moves actin to the center of the sarcomere, attaches new molecule of ATP, and is removed from actin filaments. And here we can see contraction of sarcomere, that lines they are going to the midline of the sarcomere. And here we can see sarcomere, myosin heads, they are moving actin to the center of the sarcomere, and during relaxation, that lines are going to their previous places. During contraction and relaxation, it's a cycle of muscle contraction in skeletal and cardiac muscle. And now we are going to the contraction of smooth muscles. You know that smooth muscles, they haven't usual structure of myofibrils, which are characteristic for skeletal and cardiac muscles. But let's look how contraction of smooth muscles is provided. So here we can see smooth myocytes, and they haven't myofibrils, usual myofibrils with transverse striations. But also they have contractive filaments, they have actin and myosin filaments. And here we can see dense bodies, special dense bodies, which are alike Z lines of sarcomeres. And actin filaments are attached to those dense bodies. Here we can see attachment of actin filaments. And they form this three-dimensional network inside the smooth muscle. And myosin filaments, they are depolymerized, and in the resting smooth muscles, they disappear. And only during contraction, myosin filaments, they appear, they are polymerized, and they are moving dense bodies to each other, and it results in the muscle contraction. So here we can see actin filaments, they are attached to the dense bodies, fixed parts of the smooth myocytes, and there are myosin filaments, and during contraction they move dense bodies to each other, and they move actin filaments also to each other, and it results in the contraction of smooth myocytes. And let's look, it's relaxed smooth myocyte, and its contraction, it's shortened and thickened, and relaxation of smooth myocyte. and contraction and relaxation. We cannot see contractions of the sarcomeres. We cannot distinguish any transverse striations because there is another pattern of arrangement of contractive filaments. And there are dense bodies. Here we can see them. They are attached to the submembrane complex. And here we can see actin filaments. And myosin filaments appear near them and they move polar bodies to each other. And it results in the contraction of smooth myocytes. Here we can see what happens. Polar bodies, they are located closer to each other as a result of this. And relaxation. Contraction, and during relaxation myosin filaments they disappear and it provides relaxation of smooth myocytes. And you should know that smooth myocytes they receive information, nerve impulse from the same source, so all smooth myocytes they undergo contraction at the same time.